Okay, I should just start by apologising to those interested in biotic interactions, because it's going to be another abiotic interactions talk, but I think what I'm picking up, there's clearly, certainly from a microbial perspective, we are gaining global understanding. A lot of studies around the world are showing similar patterns, so we are beginning to define, I guess, global soil biomes, and I guess it provides a baseline for further experimentation, so we can understand how different pressures affect different soil communities across the globe. I think it's clear that it's all happening. So I'm going to talk about predicting and understanding the effects of land use on soil, bacterial communities, and a few other communities over large scales. Just start with the talk outline. I'm going to talk about bacterial communities as a good indicator of change due to land use. I'm also going to talk about some of these, in more detail about some of these relationships between biodiversity change and change in soil properties. And finally talk about prediction, interpretation of indicator taxa responses, and I'm using large-scale data sets to address these issues. It's worth starting with a kind of caveat, really. When you work at a large scale, you do tend to generalize. So taxonomically, you kind of you can't go down, as we heard before, down to species level. So we're not addressing those sorts of issues. And also, what I'm not talking about generally here, we're talking about bulk soil evaluation, not specific localized rhizosphere plant-soil interactions. I think these are quite important distinctions to avoid misinterpretation. So part of the reason we're all here, as I'm sure you've heard, is due to the EcoFinders project, a large European FP7 project that's coming to an end. We've just had our final meeting on Monday and Tuesday. But this project was basically looking to study across Europe the effects of seeking to standardize methodologies for assessing soil biodiversity, looking at the effects of land use change and trying to work out if there are any consistent indicators of land use change in different sort of soil pedoclimatic scenarios. So basically a massive effort with everybody on the project coming together, everyone sampling using the same methodologies to sample and standardizing methodologies. So straight into some data. So what we have a network of sites. There was basically two elements to it. There was 5 individual sites, each one with a gradient of land use intensity. They're very different sites. We had forest sites in Sweden, grasslands in the UK, forest grassland in Italy, and also land abandonment study we heard about this morning from Vim, and some crop rotation studies in France. So clearly very different land use intensities, but what we did, we did 2 samplings, sort of spring and autumn sampling. We used various methods, TR4P, pyrosequencing to address, and quantify the impacts of these changes across Europe. And we can clearly see here, these are results of, I guess, a multivariate ANOVA. The closer to 1 it is, the stronger the effect. But for different taxa, we're seeing very strong, consistent indicators of land use effect, but it differs according to each site and what those specific management intensities were. We then went on from this to do some variation partitioning just to see, quantify the effects of the land use intensity against the sort of soil parameters. And what we see from this, it's quite a complicated plot, but for example for bacteria, you can see that actually a lot of the variation at each of these sites is due to change in soil properties. And where you get large changes due to land use intensity, it's often accompanied by large changes in soil properties. Now, I'm going to come back to change due to land use, but I just want to touch upon what we've heard about already quite a lot is, these relationships that we do get with particularly bacterial and archaeal communities with soil properties. So here we've got soil pH on the x-axis for bacteria, archaea, fungi, and I've just plotted it's the first axis from an ordination. And you can see for bacteria, we're getting, this is across the EcoFinders transect, so a wide range of sites across Europe. We're getting very strong relationships with pH. Similarly for archaea, fungi, not so much, more to do with CN ratio. Well, it's very similar really, CN ratio or pH, but these relationships are useful because it does mean that you can then, if we have global pH maps or European pH maps, we can then predict. This is a bacterial diversity map for the UK, which we've done based on the modelled relationships here. So in terms of getting towards this synthesis of global soil biodiversity, these relationships are actually really useful. We could potentially do the same for fungi and for archaea, but the models will we'll have to choose different modelling approaches. So a lot of people say, okay, well, pH, is it really pH? What about calcium? I think we might hear something about aluminium next. What is it? What is the main driver? And this is something people keep asking. And I've learned to shirk it, basically, with this. I don't think any of this would be a surprise to one of the founding pedologists. Soil pH is just one of those soil properties, as is soil biodiversity. And all these soil properties are essentially a product of climate, parent material, time. We heard about succession this morning, and relief as well, the shape of the land. So, soil biodiversity, soil properties, they're not independent variables. They are a function of the succession according to parent material and the climate. So, human impacts, humans, I guess I've substituted for organisms there. Humans do have a large potential to modify these relationships. Okay, going back to land use effects now. Okay, so we have these 5 sites across Europe. What I wanted to do was look at some old data sets. This is as part of the Ecofinders project, to say, well, okay, we've got this large-scale survey across the UK that we did back in 2007. Each square is a 1 kilometre square, and within that there's 5 random samples. So what I've tried to do is to say, okay, well, let's look at land use effects within and try and quantify constrained at the 1K level. So I've kind of looked at all the different transitions and quantified at each for each countryside survey square. It's not an ideal, I don't want to call it land use intensification, it's a land use transition. I haven't filtered for altitude yet, but so it's to gain a sort of, or try and quantify different land use transitions and the effects that they'll have on soil properties. So I ended up with 614 contrasts across the UK. And here all I've done is taken the bacterial community scores, so I guess community composition, and I've ranked each of the different land use transitions. So we've got our less intense. I had to do some ranking, again, we just heard then, ranking of different habitats, which it is kind of confusing, and sometimes it's easy, acid grassland to improved grassland, but in other contrasts, it's not really that clear what would classify as a sort of intensification of a certain land use. What you can see anyway is that, firstly, so this, as I say, I've ranked these transitions according to the effect on the bacterial community shifts that we see in these transitions. And community composition, diversity indices, you see quite a similar pattern as we'd kind of hope. And then, of course, pH, we get very similar effects as well. And, it's not so clear for CN ratio, organic matter content, but there's still some relationships here. So the context of these land use transitions is really important to determining the magnitude of the effects that we see on soil properties. This, I've just illustrated it here again. This is, again, countryside survey data. So if we have a gradient of soil pH, kind of broadly corresponds to a gradient of, more, I guess, extensive grasslands to this end, extensive habitats at this end of the gradient, going over to crops. Humans strive to get things to pH 7 so we can grow, maximize productivity. This causes a decline in organic matter and causes large changes in soil biodiversity. So that really just sums up, we can think of intensification as pushing things that way, extensification as pushing things that way. So what does this mean for indicators? Well, what we have now, this is actually old TRFLP data, but we could improve on this now with the new sequencing methods, but we have certain indicator taxa that are responding across these gradients as well, which are driving these changes we see in community composition. So here we see acidobacteria declining in red. In blue here, there's sort of alpha proteobacteria, things like bradyrhizobia, things like that. which have a more of a unimodal shape. I wonder if that's what we saw in the last slide, possibly. And then with increasing pH, you get a large increase in lots of other taxa, notably actinomycetes. We know this. So, knowing what we know about intensification pushing this way, extensification going that way, these strong relationships with soil properties, we can begin to look, okay, so I could predict that if we've got let's say an extensive habitat here, an acid grassland, and we improve it, we'd probably see an increase, we'd see a decline in acidobacteria as we move across this gradient, increase in alpha proteobacteria. Similarly, if we've got a pH 8, so calcareous grassland, and we say we've got an arable field of pH 7, we look at a transition to a more extensive habitat, so a species-rich calcareous grassland, increasing our organic matter, we could equally predict that there'll be an increase in alpha proteobacteria. That's exactly what we see. So I think we touched upon the fact that there are universal indicators of change in soil bacterial communities at least, but actually the responses or the direction of the response will change according to the nature or the habitat context of the land use transition. So here we've done a 454 survey of the southwest region in the UK. So characterised, you've got these wet, characteristic species-rich grasslands there, so they're acid. And when we look at the transition between the pristine and the intensive grasslands here, we see an increase in these bradyrhizobial taxa. And this corresponds with what I was saying before. So you're increasing the pH, decreasing organic matter, and you'll see these organisms going increasing. But similarly, when we go in a calcareous setting, when we move from on the transition from pristine intensive soils, here we get the same indicators. They're one of these groups that are just everywhere. But here, the bradyrhizobia, rather than being an indicator of intensive grasslands, are actually an indicator of the pristine grasslands. And this is all entirely predictable by knowing the context of these land use transitions across these gradients. So the relevance of these indicators, well, clearly, there are potentially, for policymakers and for monitoring purposes, there are a lot of novel new indicators we can pull out. But I think we've already heard about this really this morning, but really there's a lot of new evidence coming out that, knowing, we can confidently state now, so many people are seeing these things, knowing about some of the theories about the oligotrophic and the copiotrophic taxa, how these affect carbon cycling in soils. So we've heard about Kelly and Noah's stuff, and we've touched upon this as well, looking at specifics of carbon cycling due to different plant inputs and how it affects these abundances of these taxa and how this causes changes in soil carbon cycling. Unfortunately, I haven't got time to talk about that today. But, so conclusions and acknowledgements here. Magnitude of land use effects on soil properties and biodiversity are linked. The effect size is dependent on the nature of the transition and the habitat context. We can start to predict these effects from using this large-scale survey knowledge. And going on to indicators, yes, there's plenty of indicators there, but in terms of understanding them, it's again context dependent which direction these things are going to go. So I'd just like to acknowledge the EU Ecofinders project led by Philippe and Mark, whose work package I worked in, and all the Ecofinders partners for all the amazing sampling and the coordination that was carried out, and then the Countryside Survey team, and then the calcareous study, Richard, who I work with in CEH. And thanks for listening. Thanks.